about the general aspects of Alzheimer's disease. And I mean, if it would be that easy, yeah, we probably might have found a cure already. So today and tomorrow and the next day, we will go into more details and see that it's much more complex, the whole story. And we started yesterday we ended yesterday with the amyloid hypothesis, stating that we said we have the Aβ protein, which can be cleaved, and then eventually the Aβ-peptides, most of them with 42, or the toxic forms of 42 amino acids, will be generated. They, for some reasons, cause some stress within the cell. We have pair helical filament formation, which is tau, basically, and then we have neuronal dysfunction and later on dementia. Okay. Today we will go even more into detail of the cleavage and the processing of the ATP. And we said yesterday that the general aspect is, as you see here, but I will draw that here on the blackboard because we have to come back to that today all the time, because there are many more peptide fragments that are generated and they have different functions during the disease. So we do have the APP molecule, and the APP molecule basically sits, as we always say, within the plasma membrane. We have the non-immunogenic pathway where alpha-secretase cleaves, and we completely leave this out, okay? Because this will never cause Alzheimer's disease. This is more or less the physiological cleavage pattern, which we don't know the function. What we know is, for the pathophysiological functions, so the amylogenic pathway, that we have the so-called beta secretase, and this enzyme was first cloned, and the name was base 1. And we have the so-called gamma-secretase, and this enzyme, as you know, is the presenilin protein. When you have a mutation, you might get Alzheimer's disease with 17. So you could have all gotten if you would have had such a mutation. Right? Okay, so this is the regular processing of APP in the amylogenic pathway, and this cleavage will ultimately lead to a beta generation. And when we talk about a beta, we always start with position 1. And we say we have, for example, as we heard yesterday, Aβ 1 to 38, Aβ 1 to 40, Aβ 1 to 42. And this one here is the most toxic. Okay, so far so good. As we said yesterday, here again, Western blots. Today there will be a lot of Western blots, and I'll try to walk you through them. When alpha-secretase cleaves, we have C-terminal fragments, and we have soluble APP as alpha. And when the beta-secretase together with gamma-secretase cleaves, we will generate Aβ-peptides. Okay. So this was the basic story. Now it's getting more complicated. When you look into the brain of an Alzheimer's disease patient and you do a Western blot for the Aβ-peptides, as I've shown you before, you unfortunately do not only see these peptides here, but you see 1 to 38, 1 to 1 to 1 to 1 to 1 to, and here 2 to 42. So suddenly you get a peptide which is cleaved a little bit shorter than that. And that, for this paper here, came out in 2001. So it's in science-wise, this is ages. We know that forever, that this exists, but nobody cared. The problem is, nobody cared because base 1 is absolutely not capable of generating that peptide. But since people who are working with base they just neglect that peptide and always talk about 1 to 40, 1 to 42. In Alzheimer's disease, these two brains here are from Alzheimer's disease patients. You see there's a lot of 2 to 42, and when you look into brains of other neurodegenerative patients, there's only very little of 2 to 42. So this might be actually a marker for Alzheimer's disease, maybe. And I'll try to convince you today that there might be something to it. So base, as I said, always cleaves, if this gray shaded thing here is the Aβ-peptide, base always cleaves here in front of that aspartate and cannot cleave in front of that alanine. But maybe another enzyme is capable of doing that. And we have to find that enzyme, because there might be more to it than only base 1. So, if that is correct, we can go back to the literature again and think, are there only 2 to 40 peptides? No, absolutely not. There are other peptides. So now we've learned there is additionally, let's say, 2 to 40 or 2 to 42. Here is a paper showing that we have peptides with starting at position 3 to 40, meaning even further down cleavage here. Again, a position where base will never be able to cleave. Completely neglected forever in the Alzheimer's field. So, and this is shown here that scientists here from Ulrich Demuth, he is now at Fraunhofer Institute in Germany, has found that there are peptides that have a weird structure. They look like this, a normal Aβ-peptide, with starting at position 3, and is then cyclized. And they are so-called now pyroglutamate Aβ-peptides. So these are peptides that are generated by an unknown enzyme, position 3, then another enzyme cyclizes this, and apparently what he claims is that these peptides are even more toxic than Aβ 1 to 42. Because he claims these peptides aggregate much faster. And when something aggregates much faster, you have faster soluble oligomers, which are more toxic to neurons. So that might be a problem. So I won't tell you anything about the 3 to 40 peptides today, because I have really no expertise in doing that, but I will tell you a story about the 2 to 40 and 2 to 42 peptides. And it was a funny story because a couple of years ago, a now friend of mine contacted me. He was working on an enzyme, and what he did was he performed this kind of assay, terminal amino isotope labeling of substrates. That means nothing else than he has a cell line without one enzyme and a cell line with a certain enzyme. Then he dumps substrates or any kind of proteins on these cells, and these cells will then process, when they have that enzyme, these substrates. Then he adds a radiolabel N-terminal ligand to it, and afterwards, when he mixes these samples, can measure using mass spectrometry which proteins are cleaved and which ones are not cleaved. And then he just analyzes through data sampling, bioinformatical database research, which proteins are cleaved or not. So this way he finds new substrates for enzymes. And so I had no clue that he was doing that. He called me and said, well, here I'm working on this enzyme, you don't know me, I basically don't know you, but you're working on my substrate. And the substrate was APP. So then we started to look at what is going on with this enzyme and APP. And this is the enzyme modeled in a 3D model. Usually it should rotate, but doesn't for whatever reason. And here APP is modeled into this enzyme. So what is this enzyme? This enzyme is called meprin, metalloprotease from renal tissue. So this is always funny, these names come up when you find a protein in a certain location. So they found it obviously in the kidney. So it comes back in the name, and it's now called meprin. Well, you have that on different locations in your body as well as in your brain, but it's still an enzyme named after where they found it. It's a zinc-binding metalloprotease. It's glycosylated, and very funny, it's exclusively expressed in vertebrates. So think about yesterday. Yesterday we say APP is expressed regardless whether it's worms, Drosophila, whatever, so invertebrates, but they will never develop Alzheimer's disease. Maybe due to the fact that some enzymes that are needed are only expressed in vertebrates. Okay, but that's just pure speculation. And this enzyme acts as a dimer on the cell surface of each cell. We tested whether this enzyme is at all expressed in the brain. And we did qPCR, meaning quantitative PCR analysis. And as you can see here, most meprin is expressed by far as they found it in the kidney, right? Then there is a lot of meprin expressed in the brain in comparison to the rest of the body. So there is meprin expressed in the brain, and when we stain human brain slices for meprin, you see here in brownish neurons stained for meprin beta. Okay, so it is theoretically there where we need it. Then we started out to perform very simple experiments. He basically gave me some meprin enzyme, I gave him some APP protein, we just dumped it together in an Eppendorf tube and wanted to see what's happening. And as you can see here, this is APP without any enzymatic activity in there. But when we add meprin for 1 minute, 5 minutes, 15 minutes, you see APP is completely degraded, and you find different bands in lower sizes. So apparently meprin is capable of cleaving APP with different positions, whatever positions that may be. And we found out that these peptides are around 20 and 11 amino acids big. That would mean, with the antibody we used, that the cleavage site would be for meprin, for APP. Well, that one doesn't work. Let's try that one. Would be somewhere here or somewhere here. Okay? So far away from any beta sequence and shouldn't have anything to do with Alzheimer's disease. So then we said, okay, well, when we dump it into, if you dump an enzyme with a protein into an Eppendorf tube, you can be pretty sure it wouldn't cleave. You have so many unspecific activity of enzymes, so they will cleave. So then we said, okay, we'll try it in tissue culture, and transfected cells, either with APP alone, here, or with meprin alone, here, or with APP and meprin together, and suddenly you see this shorter bands coming up as I've shown you before. So even in cells, these bands are generated. So then you would say, okay, that's all artificial. If I blow a cell full of DNA, they will put a protein somewhere and it will be cleaved eventually. Is that true endogenously? So when you're doing really nothing to a cell or to an animal. On the left-hand side here, you see a control, 1 and 2. We have cells, we have mouse brain of either wild-type mice. So you see that APP band here, which I've shown you before. And we have a meprin knockout mouse, that band is missing. So when you don't have meprin, this band can't be generated. And when you look into brains of humans, you see normal APP up here, but you see nice bands coming up here at the size of this one. In the very beginning, we were really freaked out because we thought, oh, cool, maybe we have something which we can relate to Alzheimer's disease. But we can't at all, because we have tried here brains from control brains or MCI brains or Alzheimer's disease brains. There's really no difference. All of them have these fragments. So meprin cleaves APP up here, regardless whether you have Alzheimer's or not. Okay, so it's a physiological function. Which one? No clue. Then we thought, okay, maybe we have still something in our hand that might be worth a nice publication. Because a couple of years ago, the group from Marc Tessier-Lavigne published in Nature that APP has a completely different function in regards of Alzheimer's disease. What they said is that APP is cleaved in the way, if this is here APP, that the N-terminal part of APP up here is released from APP and then binds to a death receptor. And through this death receptor, you have cell death in neurons, and then you have Alzheimer's disease. And then we thought, hey, cool. Now we have the enzyme that cleaves up here, and therefore, when more of this peptide is generated, you should get cell death. So this is the cartoon for that again. So we thought we have APP here, we have co-expression of maprin, and then maprin would release this N-terminal part up here that would bind to the death receptor, and you should have cell death. Or maprin cleaves here and cleaves these two parts again, and therefore these two parts can't bind to the death receptor. Well, we tried so many different approaches with neurons, but we could never kill neurons with these N-terminal APP fragments. And usually it's very easy to kill neurons. When you have a primary neuronal culture, and I mean, you just shake them and they die, right? But we tried really hard to kill neurons, but we couldn't kill them with either maprin alone or with the N-terminal APP fragments or with APP alone. No way. So we don't think that the N-terminal part of APP kills neurons. That was a bummer. We were working 2 years on that and still could publish it, but not as cool as we hoped. Anyway, then we went back to the basic database and said, okay, maprin can cleave APP at the very end terminus, but maprin should be able to cleave APP at different positions close to the Aβ sequence. Because when you look at the cleavage specificity of maprin beta, you can see that they really like aspartates, which is here, or glutamates or alanates, which we all have here, right? So theoretically, maprin should be able to cleave exactly at the position where base cleaves, and should be exactly able to cleave at position 2 and 3. And then we would be back in business here for our peptides that we have seen in the brains of the Alzheimer's disease patients. So we did some basic biochemistry and performed some kinetic analysis where we analyzed the affinity of a substrate to an enzyme. So if you don't know what it is, that is basically, it just tells you the likelihood of binding of a substrate to an enzyme, and it tells you how fast the enzyme will then catalyze the cleavage of the enzyme. Okay? And it turns out that maprin beta is cleaving these short peptides that we use with the Aβ sequence, is cleaving these peptides much faster and with much higher affinity than bases do. So maprin can cleave Aβ, at least in a tissue culture tube. That doesn't mean anything. So then we said, okay, is there maybe, to most of the enzymes you have inhibitors available, and we looked whether we have an inhibitor for maprin. And indeed, we have an inhibitor which is called actinonin. And here's the structure for actinonin. But it's a terrible inhibitor. Right? It's a really bad inhibitor. It doesn't work well. It's not specific for maprin. It does do other things. But we have really nothing else. So we try to use actinonin on cells that do not overexpress maprin. So just regular cells, but they overexpress APP. And we wanted to see whether we have an effect on Aβ production at all. So you just dump actinonin on cells, and you see you have a 20% reduction in the overall Aβ production in a cell. And that is literally not related to base 1. So another good indication that maprin might be able to cleave APP. So meprin then has three predicted cleavage sites for APP at position 1, like base. 2, base can't do that, or 3, base can't do that either. So then we say, okay, we'll test that whether meprin can generate Aβ in a cell. We transfected the cells with APP and meprin and tested whether Aβ is generated. So in your first line here, we have only APP, no meprin. We do see some Aβ, which you would expect because it's generated by base. Second lane, no APP, but meprin, no Aβ, of course there's no APP. Third lane, APP meprin, and you get a lot of Aβ, right? So now we wanted to see whether that is really a product like Aβ, could be something else. So if you want to test whether you have Aβ in your tissue culture, what you do is you block gamma-secretase, for example, with an inhibitor, so-called DAPT. And when you have your cells expressing APP, meprin, and you add DAPT, Aβ is gone. So meaning this cleavage still works, that doesn't work, no Aβ. Then we tried it with actinonin, our really bad inhibitor for meprin, and you see we have less Aβ than there, but it's not really gone. So the inhibitor sucks. And this is just a control. So that was a pretty good indication again that meprin, after overexpression, can generate Aβ-peptides. So then we wanted to know is what part of meprin is doing. Meprin, as you see here, is a transmembrane enzyme, but it is released into fluids of the body and works as a soluble enzyme as well. So we added soluble enzyme to cells, and not only soluble, but soluble inactive enzyme as well. So we have an inactive form of that enzyme to see whether the soluble form is capable of generating the Aβ-peptides. So when we do that, you can see here that we have, when we add soluble meprin in different concentrations, we have no big influence on soluble APPS, but we have an influence on our N-terminal fragments, which I've introduced to you before. But the inactive meprin has no effect on these. And meprin, the soluble form, has really no big influence on Aβ generation. So from that we concluded, okay, as long as meprin is within the membrane, and APP is within the membrane, then it might act as an enzyme here and can cleave there. But as soon as it's soluble, no function on Aβ generation. So, I told you that already. Okay, so if this is correct, if we saw all that, we should be able to detect cleavages at position 1 or maybe at position 2. And here you see already something I will come back to a little bit later. You see the APP wild type sequence, KMDAE, and the Swedish sequence. So Swedish sequence meant that was a mutation in a Swedish family which resulted in Alzheimer's disease when you were 35. Right? So that was an amino acid exchange up here. So we wanted to see if meprin beta is capable of generating shortened Aβ-peptides. So since meprin is capable or likes the aspartate here, we would assume that it could cleave between the aspartate and the alanine. First, one thing that the reviewers told us to do was, which was a good idea, they said, okay, it's nice that you can generate Aβ-peptides when you overexpress meprin, but do you know whether meprin has an effect on base? So maybe you transfect a cell with meprin, and meprin then activates base, and therefore you get more Aβ-peptides. So what we had to do was we got base knockout sites. So there are two versions of base 1 and 2, and these cells do not generate any Aβ under normal circumstances. But when we transfect in meprin and APP, you get Aβ back, right? So there is no base in these cells, but we can generate Aβ-peptides with meprin, and we can inhibit it again as we have done before. So we think meprin is capable of generating Aβ-peptides even without base 1. So then we subjected our Aβ-peptides to mass spectrometry. And I'll walk you through this. So what we did here is basically, we just tested cells that only express APP and nothing else, and then we put it on mass spec, and you see, book, one peak at position beta 1 to 40. Then we used just meprin. No APP, no signal. When we have APP together with meprin, we see the 1 to 40 peak, but we see an additional peak at 2 to 40. So this now is really clear indication that meprin beta is capable of generating 2 to 40 peptides, which base will never be able to do that. And we can block that completely with our gamma-secretase inhibitor here, DAPT. And this is just a control. Then on top of that, we wanted to go a little bit deeper and said, okay, we'll do that in a different cell line. Maybe it was a cell line specific effect. Inhibited again with DAPT, no signal. And then we inhibited with a base inhibitor. So now, really no Aβ should be generated if the people are correct that base is the only enzyme that can generate Aβ-peptides. And when we have a base inhibitor, shown here, in the presence of APP and meprin, we still have the Aβ 2 to 40 peptide. And we even have 1 to 40 peptides. Only generated by meprin. This is a story that bugs us for the longest time. And from now on, I'll show you all data that is not published yet. And we are struggling so hard to sell this to the Alzheimer's community. Nobody wants this. It's completely against the stream of Alzheimer's disease research. Because Alzheimer's disease research focuses all companies, everyone, to 100% on these two enzymes, on nothing else. So if you come up with a new idea, well, you have to convince them. So I'll try to convince you now that meprin really plays a role. You can tell me afterwards whether I did or not, and if you tell me what I have to do on top of it, fine, I'll do it. Because I really want to sell that story. So the first thing we wanted to know is if this is all correct. If we're lucky, we should be able to see meprin and APP together in a complex. Right? And in a complex could mean a co-immunoprecipitation. That means basically you take an antibody, and that antibody binds to your protein of interest, and then you couple your antibody to really a bead, and together with that bead, you can pull down the protein out of any kind of solution. So, and if you're really lucky, then there are some other proteins still stuck to your protein that you have pulled down. And those proteins you can afterwards detect with another antibody. That's what we're doing. So we have a lysate. So we have in our lysate meprin and APP, or APP alone. And when we now immunoprecipitate, we come with an antibody, for example, for meprin beta. So we pull down meprin beta, have that in our tube, put that on an STS page, run a Western blot, and come then back for an APP antibody. We see here the APP band coming up, so APP was pulled down together with meprin in one complex. And when we have meprin alone, there is really nothing, right? And we could reverse that as well. So we can co-immunoprecipitate APP together with meprin, or meprin together with APP. And this is on an overexpressing level, and the reviewers told us, okay, that's not good enough, do it on an endogenous level. And that is really, I mean, we were finally able to do that, but that's, if you really publish a paper and somebody tells you that, you know that's not a nice thing to ask for. Because usually when you find something new, you don't have the antibodies to do that. You have to generate everything from scratch. And when they came up with base 1, They never did that on endogenous levels. There is no publication where they have endogenous base and APP together. No way, doesn't exist. But we had to do it, and we did it, and it worked. So, then we wanted to know where in a cell or in a tissue maprin and APP might interact. So we started to generate different APP constructs. So we have wild-type APP, where you see full-length APP, so this thing here. And at the same time, there are some C-terminal fragments generated. These are these fragments here, which are stuck in the membrane after alpha-secretase is cleaved. Then we generated a construct which we call APP-ER. It's very easy to get a protein stuck in the endoplasmic reticulum. You just add 4 amino acids, specific 4 amino acids to the C-terminal tail, and then the protein is retained in the ER. And then suddenly this protein forms dimers in the ER. But it doesn't matter, but this will never leave the ER. You don't get C-terminal fragments. It's stuck in the ER. We just used that construct to see where maprin might interact with APP. So this here now is the co-immunoprecipitation again, with all these constructs, APP, C99, so only the C-terminal part here, APP retention in the ER, or a control where nothing is transfected. And you can see in the co-immunoprecipitation that when we precipitate with maprin and come back for APP, full-length APP binds to maprin, hardly any C-terminal fragment here, but if we express C99, we see something, but when we express APP, which is retained in the ER, we have interaction. So we think maprin and APP interact already in the ER, then travel to the cell surface, and on that way, maybe Aβ is generated. Maybe. So can we visualize that? Yeah. So what we did is we used a split GFP assay. A split GFP assay is a very easy assay to use. What you do is you have the green fluorescent protein, which you can cut in half genetically, basically. Then you fuse one part to one protein and the other part to the other protein. When they get in close contact, you will have a green light, meaning you have interaction. Okay? So when we transfected these constructs, you see here a green cell, so there APP and maprin are interacting. On top of that, we wanted to know where are they interacting. So we fixed the cells and used markers, for example here for the endoplasmic reticulum, which is usually red then. But you see here already, it's all yellow, meaning when you have red and green, it becomes yellow. Right? So you have co-localization of APP and maprin in the endoplasmic. Then we looked at the Golgi. We used the Golgi marker here, a cis Golgi marker, and you have here yellow dots in a better magnification. So maprin interacts with APP in the ER and in the Golgi. Then we used an early endosomal marker. So normally APP travels from the ER, Golgi, cell surface, back to the endosome. But here, we hardly see anything. So we think the processing takes place prior to the endosomal compartment. And we think it actually happens at the cell surface, because now we performed a staining. The reviewers didn't like our assay here. They said it's very unspecific, and they are correct with that. So we performed a staining where we used an APP antibody and a maprin antibody on neurons, and you can see here that we have a surface staining. Oh, is it neurons? No, it's hex cells, sorry. A surface staining of maprin and APP at the cell surface. So they are closely, they co-localize. That doesn't mean that they directly interact. Okay, so if that is correct, we should have an effect on Aβ production, because usually APP travels to the cell surface and then afterwards is endocytosed into the endosomal compartment. The endosomal compartment, in here, 80% of Aβ is generated by base 1. That is a fact. Okay? So if we now block or inhibit the uptake of APP into the endosome, we should have less Aβ production. Right? We used a construct that is well known in the field, where you just delete 4 amino acids here, and these MPXY amino acids are a signal for a surface protein to be endocytosed into the cell. When you delete that, that protein will stick to the cell surface and will then be either further processed at the cell surface or stays there forever. And here you can see our wild-type APP or our APP with this mutation, so it can't be endocytosed anymore. Under normal conditions, you have a little bit Aβ here, but when we co-express meprin, you see a lot of Aβ, even in the presence of a base inhibitor. These APP delta and PXY constructs, they normally don't produce Aβ, which you see here. But when it's co-expressed with meprin, even they produce as much Aβ as the wild-type APP. So we do not need the endocytosis of APP for meprin to generate Aβ peptides. Okay? And meprin is nicely doing its job in the way that it generates Aβ 1 to 40 peptides, as well as 1 to 42 peptides, as well as to 2 to 42 peptides. Or these are 2 to 40 peptides, right? And these are 2 to 42 peptides. So it's generating the shorter peptides. And it's doing its job at the cell surface, and we show that by a surface biotinylation experiment. What you do there, it's again quite easy. You add biotin to cells. Biotin is a protein that binds to every protein. Right? So all the proteins at the surface are now labeled with biotin, and then you you lyse the cells, and you pull every biotin labeled protein with streptavidin down. Come with an antibody and test which protein is labeled at the cell surface. And you can see here, at the cell surface, we have APP and we have APP delta and PXY. But as soon as we have co-expressed meprin, which is at the surface as well, APP is gone and APP delta and PXY is gone. Meaning, meprin cleaves APP either at the cell surface or shortly prior of reaching the cell surface. So, we think that meprin is capable of generating Aβ peptides at the cell surface, which is completely not normal, because so far everyone believes only base can generate Aβ peptides in the pathway of the endosomal compartment. Okay, so let's think about it. Have I convinced you so far that meprin is capable of generating Aβ peptides? Yes or no? Okay? So you would all think in your brain, meprin generates Aβ peptides. Have I proven that?
Yeah, you show it. Okay.
No way! I just used tissue culture cells, right? I have not proven that you generate Aβ peptides in your brain. So, I'll try to do that after the break, okay? Okay, let's have a short break. I'm still in the business of trying to convince you that meprin might be important in Alzheimer's. Right? So far I've shown you meprin can generate Aβ from cell lines, and I'm still continuing to do so, but there are some additional aspects. So one aspect is, as I've mentioned before, meprin is capable of generating Aβ peptides when APP cannot be internalized, and is capable of generating 2 to 40 and 2 to 42 peptides, even in the presence of a base inhibitor, again. Okay? So that seems to be pretty clear. But one other aspect I haven't talked about is, so far I've always shown you results from APP wild type. So from APP that you have in your brain. Let's hope you have that in your brain, right? Because if you have APP with a mutation, as I've told you, the Swedish mutation up in front here, will result in a higher Km value for beta secretase. So the substrate binds better to beta secretase, to base basically, and you will generate more Aβ-peptides through base. Or we have a London mutation back here. When this mutation is there, it doesn't touch base at all. It just touches gamma secretase, and you have more Aβ-42 peptides, and you die with 35. Right? These were the familiar Alzheimer mutations I've introduced to you yesterday. So, does MEPRIN have a role in generating Aβ from familiar Alzheimer's disease constructs? We tested that. And here's the surprise. This is what I've shown you all along. APP wild type together with MEPRIN, Aβ 1 to 40, 2 to 40. APP Swedish together with MEPRIN. No 2 to 40. So, is that a bummer or not? For me it was fantastic. Because what this might tell us, I'm not saying it tells us, it might tell us a difference between sporadic Alzheimer's disease and familiar Alzheimer's disease. So if you have familiar Alzheimer's disease, you have a mutation in the Swedish, you have a Swedish mutation in your APP gene. You of course generate a lot of Aβ due to base. And when you produce a lot of Aβ, yeah, you will get Alzheimer's disease earlier. No question about it. But under wild-type conditions, maybe MEPRIN is generating all the time some 2 to 40 peptide, and that at the end is the reason that Aβ aggregates faster and you get Alzheimer's disease. So then we should find that in sporadic cases. We did, I showed you that in the very beginning, right? So maybe there's something to it. So we don't see that in base, in Swedish mutations, but we see 2 to 40 again when we have the London mutation. The London mutation was the mutation that only affects gamma secretase. So down here, so the mutation up here is not affected, right? So MEPRIN only cleaves here. The mutation down here shouldn't make any difference whatsoever. And again, when we have a double mutation, again the Swedish mutation up here is in there, as well as the London mutation, we still have, you can't see that very well, but you still see nothing, right? There is no 2 to 40 peptide here as soon as we have a Swedish mutation. So, what does this tell us? This tells us I am in trouble. Because when you think about Alzheimer's disease research, for many, many years, we are using transgenic animals to mimic Alzheimer's disease in mice. And mimicking Alzheimer's disease in mice means we have to have plaques in mice, as in AD patients, and you only generate plaques in mice when you have a lot of Aβ-peptides. And you can get only a lot of Aβ-peptides when you use a human APP with a Swedish mutation in a mouse. So all the mouse models that are useful have APP Sweden here. So when we now want to look for MEPRIN activity in these mice, that's useless. They don't work. So what we have to do now, and what we're doing at the moment, we have to use mice either with wild-type APP or with the London mutation alone to see whether they have an effect on plaque pathology. We will do that, but with these animals, with these mutations, you wait for 3-6 months, you have a pathology in the mouse, right? With these here, you might not see anything at all. Maybe after 12 months or something. With them, 8 months, 9 months, maybe. So what that means is, for you it shouldn't matter at all, you don't care. But when you, as soon as you are a PI and you run a lab, I don't know how it is here in St. Petersburg, but in Mainz I pay per mouse €2 a day. Oh, sorry, €2 per mouse a week. So now add that up, how many mice you need for almost 2 years to perform these experiments, right? Money eats you up. It's crazy. So if you can do those experiments here cheaper, I'm happy to cooperate, right? No, forget it. So we will do that, but we can't do that at the moment. Well, we are doing it at the moment. We will not have the results right away, let's say that way. But this is very interesting. So MapPrint is only capable of generating shorter Aβ-peptides when you do not have a mutation close to the beta-secretase cleavage site. So, and we analyzed that in more detail with these cleavage specificity plots, and it turned out that is really correct. When you change something around here, MapPrint doesn't cleave as well anymore. So, is that always the case? And yesterday was, I think you asked me that question, is there any protective mutation in the world that protects you from Alzheimer's disease? Yes, there is. This one, APP-A673T. That mutation was found in Iceland, and apparently these people develop Alzheimer's disease much later, if at all. Because what they found was when they used these constructs in a base peptide cleavage assay, they saw that base is not cleaving as good anymore. Therefore, the people generate less Aβ, people get much later Alzheimer's disease. So, but I claim, if you alter amino acids around the beta-secretase cleavage site, you should have an effect on MapPrint, right? So here we are changing again some, at this position here, an amino acid, so we should have an effect on MapPrint, maybe. And we do. So here you see regular APP wild type expression, here with MapPrint, here with base as a control, and you see 1 to 40, 2 to 40, and here even some 2 to 42. When we have the protective mutation in there, we see less Aβ generated compared to wild type, and even less 2 to 40. We think, but we're not sure yet, that this faint band here is 2 to 40, but with a threonine, and therefore it runs a little bit different in this gel system. But even if either it's not or it is, doesn't matter. If it is, you see this here is clearly less than the one, right? So theoretically, MapPrint would be, or the cleavage by MapPrint would be protected through this mutation as well. And this is a quantification. So another good indication that MapPrint plays a role. So now we know MapPrint generates truncated Aβ-peptides. But do these truncated Aβ-peptides have anything to do with pathology? Pathology would mean we need aggregation of Aβ-peptides. Under normal circumstances, when you aggregate Aβ-peptides, and some of you might know that much better than me, this happens. The blue line here, Aβ aggregates over time and plateaus at some point in time. And Aβ means Aβ 1 to 40. But if you use Aβ 2 to 40, you have a very fast aggregation, and then it plateaus out. And this is just so a very fast aggregation of Aβ 2 to 40. So this would mean a lot of Aβ 42 would aggregate very fast, but you don't have a lot of Aβ 42 in the brain. You have very little Aβ 42 in the brain. So could it still make a difference? So therefore, we performed a different experiment and said, okay, maybe even if we have very little Aβ 2 to 40, maybe this is a kind of a seed for a beta 1 to 40 to attach to and therefore aggregate faster. Right? So what you see here is, again, just a beta 1 to 40 in green, regular aggregation curve. But when you put, the red one, it's a regular aggregation curve. And the green one, when you put just a little tiny bit of Aβ 2 to 40 to 1 to 40, the 1 to 40 aggregates very fast and plateaus off. So the 2 to 40 could indeed be a seed for Aβ-peptides to attach to and then aggregate even faster. So that would bring us back to the so-called amyloid hypothesis that aggregated Aβ is either deposited into the plaques or as a soluble form is toxic somehow to neurons. So that would mean through meprin we have a little bit faster aggregation, and at some point in time when you have enough 240, you might have faster aggregation and then conversion to Alzheimer's disease. So do we have more meprin than in a brain of an Alzheimer's disease patient? We started to look for that with quantitative RT-PCR, and how this works here is the following. So this actually means you have more meprin here in the Alzheimer's disease brain than in the non-demented brain. So now you would say, wait a minute, this one is higher than that one, right? So how this works is the following. When you do a PCR, you amplify DNA, and you always count the rounds that you amplify. And here, you need much less to get a signal than there. So there is more to start out with, right? So that's the reason, the way we plotted here. You have more meprin RNA in the brain of an AD patient than in a non-demented patient. So first indication, meprin has a role in Alzheimer's disease. Then, and this was hard, and it's really ugly. So normally when a PhD student or postdoc comes to me and shows me such a plot, I'll tell him, okay, go back, do it again, come back, and it's nice and clean. But we just had to generate a new antibody, and the antibody is really terrible, but it's doing its job. So we can actually detect now on endogenous levels in human brains meprin beta, and what you can see here is that you have more on protein levels, meprin beta, in Alzheimer's disease brains than in controls. And we have now measured from 20 brains, and it's always the same. And this is not only true on protein levels. We did stainings of AD brains versus control brains, and I think now we're over 30 now. And the left-hand side is Alzheimer's disease, right-hand side is age-matched, non-demented control. So on top here is meprin staining, and you see the brown dots again in neurons, and here only very little under control conditions. That this is really an AD case is highlighted here through the plaques that you find there. This is here, and these are two different stains for neurons, and but it just tells you that this is really an Alzheimer's disease brain, and in the controls there's hardly any meprin. We have more stains for AD and for controls, and when we quantify, these are only, I think, from 20 brains, now we're over 30, and this still holds. You have a dramatic increase in meprin expression in Alzheimer's disease brains of sporadic patients, right? So these are all sporadic patients, meaning it cleaves only wild-type ATP. So do we find then more Aβ 2 to 40 in the brains of Alzheimer's disease patients? So here we are, it's still puzzling. We are not there where we want to be. So down here again, our stainings. This is Alzheimer's disease, this is control. Here we even included some tau staining for Alzheimer's disease, but you have a lot of meprin staining in Alzheimer's disease, some in controls. So in Alzheimer's disease brains, we have some 2 to 40, but unfortunately we find that in the controls as well. So this might mean two things. Either it means meprin has nothing to do with Alzheimer's disease. Fine. Okay, possible. The other thing, which I have shown here, is something else which you should think of when you look for Alzheimer's disease brains. You not only look for Aβ 1 to 40, you always look to 1 to 42, which is the toxic form. And usually a lot of 1 to 42 tells you Alzheimer's disease brain, right? This one here, the control, has a lot of 1 to 42. So I would predict, I can't be sure, but I would predict this fellow here would have gotten Alzheimer's pretty soon, right? But he died earlier. So therefore, he's not an Alzheimer's disease brain, unfortunately. So we have to now extend our study on more and more brains. We have now, I don't know, 10 to 15 brains checked, and it looks pretty good. But to have a perfect statistics, we have to do more and more and more, but it's really hard to get all the brains that are nicely documented to prove that we have a correlation. But then again, what is the correlation good for? Right? If I can prove to you that we have high heparin beta and high Aβ 2 to 40 in Alzheimer's disease brains, and less in controls, that's a correlation. The correlation is we have, in 1989, we had a lot of storks and a lot of children. That's a correlation, right? Does it mean anything to do with each other? No, right? It's just correlation. But we are trying to get to those numbers that we can show that there might be something to it. But I can only present you the final proof when we have the animals. So we have generated animals, either heparin knockout animals, so these animals should basically never get Alzheimer's disease, or should never get plaques, or very late. That's really bad, because what kind of result is that? I mean, you cross animals to show that they don't get Alzheimer's, that they don't get plaques. You wait for 2 years, then the mouse dies anyway, right? So, that's a negative result. Somebody will tell you, well, you should have waited longer. Yeah, sorry. Lifetime of a mouse is restricted. So then, the other way around, we tried. We made a heparin transgenic animal, meaning that the animal was producing a human transgene of heparin in the brain and was expressing it even stronger. What happened? The animals died pretty fast. We even couldn't propagate them at all, because when you have an enzyme overactive in a cell or at the cell surface, it cleaves pretty much everything away. And heparin has not only one substrate, it has 50 substrates at least. And when the enzyme is overactive, you have a problem. So those animals didn't survive at all. So what we're now doing is, well, we get smarter every day. We generated an animal where we can induce the heparin after or whenever we want to induce it, right? So we put it in a locus, in a specific region in the brain, and then we can turn it on or off whenever we want to. So when those animals are finally there, then we will cross them into an APP mouse model and hope to generate plaques and everything faster. But I think we are 2 years away from that. So I still, to be honest, cannot convince you that heparin plays a role in Alzheimer's disease. But what I hope, what I could have done is to raise some interest that there is something else than base or gamma-secretase. There is surely something else, but whether heparin plays a definite role in sporadic Alzheimer's disease, I am not so sure. Okay, let me summarize what I've talked to you today. So first, I showed you that heparin can process APP in vivo. I showed that with the mice, the knockout mice. So heparin can cleave up here in vivo. I could show that. Heparin levels are increased in Alzheimer's disease brain. We've shown that by quantitative PCR on protein levels and in stainings within the brain. Heparin can produce Aβ in the absence of base, at least in cells. It interacts with APP prior to the cell surface and maybe at the cell surface, and it generates 2 to 40 peptides, preferentially from wild-type APP and not from the Swedish mutation. And we think this all happens in this pathway from the ER to the Golgi to the cell surface. There heparin might interact and cleave APP and therefore generate Aβ up here, while it's normally processed down here. So a little 2 to 40 is maybe generated up here. But I always have to say maybe, since we cannot prove completely that we are correct. So this time I'd like to acknowledge some of my co-workers, my PhD students who actually did all that. And that were here, Jessica Dean, she now left, she works in the company now. Simone Isbert, she's not on that picture even anymore, she works in a company now as well. And Caroline Schönherr, she's still in the lab finishing her PhD work. And probably she will go to a company afterwards also. I don't know, they all want to go to companies and earn a lot of money, I don't know why. Science is so much fun at university, so if you like it, stay there. You won't find freedom as you will find in the university at all. But anyway, these are the collaborators and some funding organizations. And I hope I didn't stress you too much, because now it was getting really into molecular details. And I was glad that you were here, and I'm open for questions. Thank you. Harder stuff than yesterday.
I don't know. It's relevant. It's all about the Aβ-peptides, that it plays a role in aggregation of Aβ-peptides.
So what you're asking me about the aggregation of 2 to 40 and 1 to 40, and there you lost me.
It's just aggregation, you mean in all the cells, or aggregation in those plaques?
Outside of the cell, I would assume. We haven't shown that. So this is in a test tube, right? But I would think when Aβ is released into the fluids of the brain, let's say that, eventually Aβ aggregates, and then it is somehow binding to neurons and it's toxic. How? I don't know, right? But when 2 to 40 is in that mix, it aggregates faster. I think even in the brain, right?
So it's not that toxic if it doesn't aggregate.
Correct. If it's not aggregated, it's probably not as toxic. So we need aggregates. That might be the reason why you don't get Alzheimer's now. No, I mean you're probably not aggregating as much Aβ, or you can clear it. We'll talk about it in 2 days. But if it were aggregating in your brain now, I would predict that you will get Alzheimer's. Yeah.
Okay. And you actually told a lot about 2 to 40 or 2 to 42 peptides. And have you got any information on the 3 to 40 peptides from other groups? Or?
Yeah, other groups have shown that, and they're working really hard on it. And they have shown very nicely that when you, for example, overexpress the enzyme that cyclizes the Aβ-peptide, you get very strong plaques. You need only a very little bit of the 3 to 40 peptide to have even more faster aggregation. They have even made mice. That looks all really good in the animals. So the 3 to 40 is probably as toxic as the 2 to 40, or maybe more, I don't know, but it is certainly a problem.
And does metron play a role in its generation at all?
Good question. Will you cut that out? Good. The reason why I'm saying that, it's a sad story.